So we are a computational biology group, and we are studying gene and epigenome regulation during cancer formation. So lots of the processes that are involved are very complex, and they include both transcriptional regulatory processes as well as changes in the epigenome and the 3D structure of chromatin. So transcriptional regulation is mainly controlled by these proteins called transcription factors, which recognize very specific DNA elements and bind to them and turn genes on or off. So Dora, who's a postdoc in my lab, and who comes from a computer science background and is turning into a computational genomicist, is looking at this transcriptional regulation in cancer. She's trying to identify targets of these transcription factors during different carcinogenic processes, and she's also looking at changes in the epigenome. So the epigenome consists of modifications that are not part of the DNA, and these are usually modifications in histone proteins, which DNA is wrapped up in. There are about 80 of these histone modifications, and there's a very complex histone code underlying the genetic code. Dora is developing methods and tools and data resources to understand these processes. So in addition to the transcriptional regulatory process, there are changes in DNA structure. So as I said before, DNA is pooled around these histone proteins, which form complexes called nucleosomes. And looking at their 3D and 2D structure and interactions is part of the research team that Joanna is involved in. So Joanna is a PhD student who's a mathematician and a computational biologist, and she's using her skill set, bringing in both mathematics as well as artificial intelligence-based methods to understand 3D structure and interactions of chromatin. So we use lots of different data sets, and most of them come from collaborations with experimentalists within the building or externally. External collaborators include those local as well as international ones. And by interacting with experimentalists and designing experiments and coming up with methods to understand and analyze these data sets, we are hoping we will understand some of the complex rules underlying carcinogenesis.